All right. Good afternoon, everyone. We're going to give it a minute, let everybody come on in. We are expecting quite a few people today, so we're just going to give it a moment. I know it always takes a couple of minutes for people's connections to work. So while you're joining us, let me just share my screen. And let me just do a couple of other things. Open up, see who's here today. Recognize a lot of familiars. Okay, one of the things I am going to do, if you need our closed captioning, our live captioning today, I just put that in the chat box. So welcome everyone. I do recognize a lot of familiar names coming on in. I'm glad you're back with us today. If you're new, we like to start by welcoming everyone. So please open up your chat box and tell us who you are and where you are from. I see Sage is joining us from Monson, Maine. Fantastic. Who else is with us today? Just find your chat box. Tell us who you are. Hi, Amelia from Greensboro. I know there's more than 2 people here today because I can see the participant list. KL, welcome back. KL is one of our regulars from Maryland, and Fuzzy's back again from Vermont, and Leo's back again from Greensboro. So nice that you all come each and every week. I know there's a couple of new people today. I do recognize a few names. And our presenter for next week is joining us. She wants to see how this all works. So who else is with us today? Let's give a welcome. Just find your chat box and go on in and introduce yourself. While you are doing that, I'm going to remind everyone of our protocols here in Zoom. And I also forgot to introduce myself. My apologies. For those of you who don't know me, my name is Lauren Traster. I am the 4-H Teen and Leadership Program Coordinator, and I coordinate all of these teen science cafes, bring in our scientists to present their amazing research so that you all can learn from them. So when we are here in our teen science cafes, all of you are muted, but that does not mean we do not want to hear from you. There are 2 ways that you can participate. One is through the chat box, which many of you have done already. And again, if you haven't introduced yourself, please open up your chat box, tell us who you are and where you are from. So the chat box, we use the chat box to communicate back and forth with the presenter, share your thoughts, answer questions that the presenter might have of you. We have a bunch of poll questions, so you'll put your answers in there. And the chat box is really to stay on topic. It's not to be, going off on tangents with others that are here, because we really don't want the chat box to be distracting. Your questions for the presenter are going to go into the Q&A box. So that's a separate item on the menu bar. And when you go into the Q&A box, you'll see there's an option. You can see a thumbs up, and that means if you like someone else's question, maybe they had the same question you had, you can click on the thumbs up and it just upvotes a question, lets us know more people are interested in that question, and it moves it up the ranks. Really, in a way, doesn't matter because we answer every single question that does come in. We like to have your questions. Presenters love to know what it is you want to know, and they're more than happy to answer every question that comes on in. So we just ask that you're courteous and respectful to everyone. Again, don't create any distractions with the chat box. Just be engaged, participate fully. I know today's topic is going to be really interesting. So before we get to today's topic, I do just want to remind you all that we have a whole series of the spring series of our Teen Science Cafe. So every Wednesday at this time, all the way into June, we have a cafe scheduled, and you can go to the website listed and see what every topic is and register. We have a really cool opportunity for teens. This is a Vermont only opportunity because it's a full scholarship, but the National 4-H has a program called Citizenship Washington Focus. Normally, it's a program where you go to Washington, D.C. and you stay at the National 4-H Center for a week and tour D.C. and participate in workshops all about citizenship and civic engagement. But obviously with the pandemic, we can't do that, but they're offering a virtual program, and it's going to be once a week for 5 weeks starting in June. And we're offering 8 full scholarships to participate in that program. So if you are someone that's really interested in civic engagement, government, learning how our government works, learning how, what goes on in DC, they're going to have virtual tours and some really cool programming, you might want to apply for this program. So you can check out the application at the website that's here. And I will say we are currently really actively working to put some in-person programming together for this summer. So that's going to be announced soon at this website. So you're all are on my list if you're part of any program, but just kind of be on the lookout because hopefully we'll be posting soon some in-person, yay, programming for this summer. So let's get to today's presentation. If I can advance my slide. So today our cafe is on the foundations of neural development, and our presenter is Hannah Boyd. Hannah is a current PhD student in biology at the University of Vermont. She received her undergraduate degree from the University of Massachusetts at Amherst in psychology and had a focus in neuroscience. When she was an undergrad, she researched auditory processing and learning in songbirds. And now at UVM, she's studying how glia function to support neuronal development with the fruit fly nervous system. So really fascinating. So let's all welcome Hannah to the Teen Science Cafe. Thank you for joining us, Hannah.
Thank you guys for having me. I'm really excited to share with you all my research. So I'll just start sharing my screen and we can get started. Okay. Oh, sorry, just trying to get. Okay. So I have the chat open, so I'm happy to take you guys' questions as we go along, and then there should be some more time at the end for any that you guys have. And even though we're talking about neurodevelopment today, I'm more than happy to try to answer any of your science questions that you might have. Or if you have questions about what it's like to be a researcher or how you're able to kind of get started with that. Some places even have opportunities for high school students to work in labs and get started in research. So I'm happy to address some of those as well. So I'm just going to start talking to you guys a little bit about who I am and my background. So I grew up in a really small town in Western Mass. And my high school, therefore, was also really small. And we had limited access to science education. And I was also a first-generation college student. So applying to college with the desire to want to work in and major in a science field was a little bit overwhelming. And I knew from my high school classes that I was generally really interested in biology. And at the time, I assumed that people who were interested in science and who also were interested in kind of helping people should become doctors or some type of healthcare professional. Now I know that there's tons of careers that you can have with an interest in science. And that can include anything from being a researcher to a teacher. You can work in government, science communication, healthcare, technology, even illustration and science art is really, really cool. And there's just so many more other careers. So one of my career goals is actually to work with young people to help them figure out what career and paths are right for them. So if any of you are feeling a bit lost or overwhelmed with the idea of applying to college or picking a direction to go in with what you want to study, I've totally been there, and I'm more than happy to talk to any of you if you want some advice at all. So I have my email on this slide, and I'll also put my email on the very last slide, and feel free to reach out or ask a question towards the end of the talk. So after high school, I ended up accepting an offer from the University of Massachusetts at Amherst, and I entered as a general biology student. And through coursework, I ended up becoming really interested in the brain. So I majored in psychology, and that with a focus in neuroscience. So how did I end up in research? And as I said, I went to college. I thought I wanted to be a doctor. So to kind of build up this application, I wanted to look for different types of opportunities that would look good on a med school application. So I just started emailing local doctors' offices, including general practices, sleep clinics, homeopathic medicine shops, rehab facilities, looking for someone who is willing to let me be involved in their practice. And this is something that you guys could all totally do. I definitely has a lot of dead ends for just kind of cold emailing people, but I had a few that got back to me, and that's how I got my first research experience. So if there's anything that you guys are interested in, it's great to try to get like an opportunity to try that out before you kind of commit to it, you know, as you get older. I've learned a lot by doing different things and then realizing I don't like them. So it kind of helps to narrow down the list of where I want to be eventually. But the person who got back to me was a clinical psychologist. The person who got back to me was a clinical psychologist, and her name was Dr. Amanda Roberts. And she was in need of assistant to help perform a study on EMDR therapy. So EMDR therapy uses bilateral stimulation of the visual field or the auditory field. So providing a stimulus to your left and right side repetitively. And right here, you can see an example of what a visual stimulus would look like. And as this stimulation is occurring, a therapist also initiates talk therapy. So usually this is only applied in a one-on-one fashion, but we were really interested in trying to incorporate this within group therapy. So most of my job during this project was just preparing test packages, recording survey results, contacting participants, and that sort of thing. And I was really excited when we got our results back and we were able to see that patients reported feeling better after only having about 3 sessions. So what really drew me into wanting to continue on in research was this scientific experimentation leading to the direct improvement of people's lives. And I realized in that moment that you don't have to just be a doctor or healthcare worker to help people, but you can also be a researcher. So we're not doing like pop-up poll questions this time, but feel free to write in the chat for what kind of people do you think would benefit from EMDR therapy? And you're welcome to choose one of these answers, or if you have a different answer in mind as well.
So as you read through and think about your answer, what kind of people do you think would benefit from EMDR therapy? A, people dealing with a chronic illness. B, people who have lost a loved one. C, people with depression. D, people with anxiety. Or E, all of the above. Seeing a bunch of E's coming in.
Yeah, that's great, guys. So exactly, like there's no right answer to this question. Like anyone who could benefit from talk therapy could absolutely benefit from combining that with EMDR therapy. So that's great. So once that study ended up being over and it went out to be published, I sought out more research opportunities. And this is when I turned to my home department at UMass of psychology. And I joined a cognitive psychology lab run by Dr. Andrew Cohen. And that was interested in the decision-making process that's involved in gambling. And we studied how culture, emotion, sleep, and time all influence your judgment decision during a gambling scenario. So as a research assistant here, I was in charge of overseeing human participants during experimental trial blocks, and I was exposed to the first time to scientific literature and how you can dive into past studies and gain a sense in building blocks for future work that needs to be done in the field. And at this point in college, I started to take neuroscience courses, and I became really infatuated with the anatomy and development of behavior and of the nervous system. And I wanted to get a more hands-on experience working directly with brains. So I ended up joining a neuroscience lab. And that lab is called the Healy Lab. And it was one of my favorite experiences during undergrad. And I studied auditory processing in songbirds and the types of signaling mechanisms that these cells are able to use to communicate. And birds learn to sing in a very similar way to the way that humans learn to speak. They undergo a babbling stage when feedback from their parents is really crucial for their ability to replicate songs and sounds. So most of my experiments depended on me staining brain slices with fluorescent markers that are able to tell you the different proteins and properties that that cell has. And after I graduated, I ended up working in the lab for another year as a lab manager. So I picked up some more managerial duties like ordering supplies and animal care and keeping up with different stocks in the lab. And I was also kept doing my own research, and I moved into some more like behavioral trials and classical conditioning, which was really interesting. And I was really lucky to have found a super amazingly supportive lab who provided me the environment to fall in love with science. And I ended up deciding to apply to graduate school, which is where I am today. And I'm going to talk to you guys about some of my actual research at the end of the talk. But this is kind of where I became, now I'm really interested in the study of neurodevelopment. So again, feel free to go in the chat. I'd love to hear you guys' thoughts, especially if you have suggestions other than these. But I'm just wondering, what kind of animal models do you think could be really useful to study communication? I know I mentioned birds, some A's and some E's. Awesome. Yeah, whales and dolphins are able to communicate with each other with complex systems of like whistles and different other sounds they make. Insects also communicate with each other a lot. If you've ever seen bees, they do like a vibration that allows them to communicate. Rodents actually use ultrasonic vocalization, so we can't hear them, but they can make like chirps to each other from long distances. Frogs communicate to each other. I'm sure you guys have started to hear the spring peepers outside trying to tell each other where they are, so they're able to find mates. And bacteria use lots of chemical cues to be able to communicate to other bacteria and organisms about where they are. And that's one way that our white blood cells are actually able to target them and find them. So what even is a brain and why is it so important? Our brains are the central processing unit of our bodies, and they allow us to think, to solve problems, to have personality, communicate, behave, react to stimuli, and so much more. Basically, anything you can do is because of your brain. So since the brain is able to oversee and accomplish such a broad range of tasks, the field of neuroscience has been broken down into a diverse and interdisciplinary web of research questions and researchers who have dedicated their lives to answering them. So many researchers who consider themselves a neuroscientist also consider themselves part of a smaller study area as well. There's just too many like big, broad questions to ask about the brain. So in science, we try to chip away at a larger question by first asking some smaller ones and then trying to piece all of that information together. And some of these branches of neuroscience include neuroethology, which is the study of behavior, neuroendocrinology, the study of the hormones in the brain. Some people are interested in cognitive neuroscience, so more complex thought, and some people are really interested in circadian rhythms and sleep. And some people are just really interested in the nitty-gritty like regulation of brain chemistry and proteins. And I'm going to talk to you about one of my favorite areas of neuroscience today, which is how the nervous system develops, and that's neurodevelopment. So just kind of wondering, for any of you out there that are interested in neuroscience, what are kind of your favorite branches or any that really spark your interest? It could be behavior, it could be learning and memory, or addiction and stress, sleep and body rhythms, cellular and molecular neuroscience, or there's so many other realms that I haven't even mentioned that I'm wondering if you guys have heard of before. See some learning and memory, that's great. Addiction and stress, that's a huge realm and has a lot of different applications to drug design and things like that. Is there any other ones out there? Some behavior, I love behavior, it's super interesting. And all different types of species have different behaviors that they use to communicate. That's great, guys. So now I'm going to get into neurodevelopment. So neurodevelopment is the study of how the brain forms, starting from an embryo all the way into adulthood. And this includes both the regulation of cell growth and division, as well as how cells are able to migrate to their final destination and form these really interconnected connections with other cells. It includes the development of behaviors that are innate, meaning you have them at birth. So this is what allows you to be born and know how to suck and how to grasp things. So you're able to be able to start feeding. And at birth, your brain is actually born with almost all the neurons you're going to have for your entire life. So development has to be a very strict and characteristic process to make sure that every part of your brain is properly formed before birth. And despite neurons being produced in fixed numbers, their ability to survive throughout your life and be able to make different connections to other neurons is really dynamic, meaning that they make and get rid of connections throughout your life. And that's what basically allows you to be able to learn throughout your life, is these connections being made, not the production of more neurons. And before we get into kind of the more specifics of neurodevelopment and the process of that, I just wanted to give you a quick neuroanatomy recap. So the main reason brains are so specialized is because they are filled with these neurons. And neurons are a really unique cell type that specialize in communication. So their job is to send and receive information along their network, kind of like a game of telephone. And the structure of different types of neurons are dependent on what kind of information that neuron is going to be passing along, or if it's a sensory neuron, what kind of information it's receiving from the environment. But in general, their structure can be broken down into the components that you see in this picture. So on the left-hand side of the cell is all of these processes that are sticking out from the cell body, and these are called dendrites. And this is where the cell is receiving communication from the previous cell in the system. And the dendrites are attached to the cell body, which is referred to as the soma. And the cell body is part of the cell that you're probably most familiar with. This is where the cell stores its DNA. It's able to create energy and different types of products that the cell makes. And then you have the axon that comes off of the cell body, and this is where electrical communication is happening within the neuron. And it's covered in this like fatty substance, which is called myelin. This basically just insulates the neuron both for protection and to increase the speed of that electrical signal. And at the end of the axon is the axon terminal, and this is where all the terminal boutons are. And this is where all of the chemical communication is going to be released between different types of neurons, and those are called neurotransmitters. So neurons use both electrical and chemical means of communication, and to start a postsynaptic cell, so this is a cell that is receiving the signal, gets a chemical cue from the presynaptic cell, which is the cell sending the signal. So the chemicals are called neurotransmitters, and they bind to receptors on this postsynaptic cell. And these signals come in two main kinds. They're either excitatory or they are inhibitory. So as you can imagine, the cell is kind of taking in all of these signals and it's summing them up of whether or not it should pass on its communication. And if there are enough excitatory signals, then the cell fires, meaning it starts to communicate to the next neuron. And this is an all or nothing response. So once it commits to firing, it is going to fire and send a signal to the next cell in the pathway. And at this point, once it's firing, the cell uses an electrical communication throughout its axon, and there's all these different channels on its membrane that allows for this signal to be passed down the length of it. And at the very end of the cell, there are these like little pockets of chemicals, those neurotransmitters, and they're waiting to be released. And once they receive that electrical stimulation, they're able to open up and they send those chemicals to the next cell where it will bind and to the dendrites and be able to process the signals all over again. So now you can imagine this happening multiple times a second throughout billions of neurons in your brain. So these cell-to-cell communications link up to form pathways, which frequently signal together. And some of these pathways form brain regions, which focus on interpreting specific tasks, like moving your body or doing what I'm doing and talking to you guys all now, or storing memory. And I hope you can understand just how complicated and intertwined these different communications can be. And that's one reason that neuroscientists try to study specific questions in areas within neuroscience. So then I would be remiss if I didn't mention to you guys as well about glial cells. So besides neurons, your brain is filled with other support cells that work to support neuron functioning, and they're called glial cells. And without them, neurons wouldn't be able to survive. So I like to think of neurons kind of as like senators or representatives, like leaders within their system. And they're making all of the important decisions and they're communicating to each other to make different types of decisions and enact change upon your body. But they wouldn't be able to function without these other people who work behind the scenes to make sure they're taken care of, right? Like, so we have people that are making sure that they're getting the correct information and they're fact checking them. They have people that are making sure that they had lunch today and cleaning their office once they leave and all of these things like that. So pictured here are a few different types of glial cells. And there's tons of other kinds as well, but these are kind of the main classes. So you can see an oligodendrocyte, which is what is wrapping the fat, which is wrapping that fatty like protective lining that I told you guys about called myelin. And oligodendrocytes function in the central nervous system, and then you basically have a really similar cell type that's doing the same job in your peripheral nervous system. So that's all of the nerves in your body versus the central nervous system is just your brain and spinal cord. And that cell type is called Schwann cells. And then there's also astrocytes, which serve as a mediator for nutrient exchange from your blood vessels. So blood has to be separated from your neurons because it can actually be toxic to it. So you have this like cell intermediate that's helping to move those nutrients and oxygen from your blood to the neurons. And they're also really important in injury response. So for anyone that has like a brain injury, they kind of work up to like make some scar tissue and plug up kind of where the damage was to protect the rest of the neurons. And then we also have microglia, which remove debris such as like dead cells or pathogens. They can function, they have like immune system functions, and they kind of engulf all of this debris and they can break it down into pieces to be excreted from your system. So I'm wondering if you guys can tell me from what I just mentioned, what are the ways that neurons are able to communicate? Can they communicate chemically, electrically, through temperature, using intermediate cell types, or both chemically and electrically? Okay, I'm seeing a bunch of E's. And that is great, guys. That's totally correct. So as we said, they're able to communicate between cells chemically, and then within the cell itself, it sends an electrical signal. So that's awesome. And I have one more question for you guys about why are glial cells important? So do they keep neurons from communicating? Do they take care of neurons to make sure all their needs are met? Are they kind of backup cells for neurons? So when neurons die, a glial cell can become a neuron. Are they undifferentiated neurons, meaning they develop into being a neuron, or they connect your brain to your spinal cord? Okay, I'm seeing some B's so far. And that is great. They are exactly as you guys are saying, and you're listening so great. So thanks for that. They are working to take care of the neurons and any needs they have, like that analogy I gave you with the senators and representatives and all of their kind of support staff. So now that you guys are familiar with some of the fundamentals, I'm going to walk you through brain development. And I've become so fascinated by the idea that we start off as a single cell and then we're able to develop into such a complex organism. Like being a human, are able to do so many more tasks than even other organisms that we interact with every day. So I was also so interested at how these cells know what kind of cell type to be, because there's so many different kinds within our brain and they all have different jobs. So like what is able to tell them what to actually end up being, and happens when there's different developmental disruptions. You probably know people or have heard of different disorders that happen due to problems in neurodevelopment, like people that have spina bifida, or people that have other types of like brain functioning disorders, or even like the groundwork that's laid during neurodevelopment can lead people to have things like dyslexia and different, or anxiety when they get older. So development starts with an egg and a sperm coming together to produce a fertilized egg, and that stage is called a zygote. And an organism doesn't stay a single cell for very long though, because divisions start occurring once every 12 to 24 hours. And a zygote is a large cell for its size. So the first few divisions, the cell doesn't acquire an increase in overall mass, but there's an increase in the number of cells within that mass and the amount of DNA. And you can imagine that if every 12 hours the cells duplicate, you're very quickly going to increase in cell size and complexity and start to see different structures forming. So as the cells divide, they form a cluster that has a fluid-filled hole in the middle, and this is called a blastula. And at this point, the mass implants itself into the mother's uterine wall, and this implantation allows the embryo to get the support from mom and develop connections to her body, and able to start delivering essential things like oxygen and nutrients and being able to excrete waste. So the first recognizable adult structure that forms is the gastric system. And this step in development is similarly named gastrulation. And during gastrulation, a hollow tube begins to form with an opening on one end. And one opening is going to become the mouth, and the other opening is going to become the anus. So you start to develop kind of your digestive tract right away. And then at this point, they start to form 3 distinct tissue layers. And these tissue layers are super important for the building blocks for the rest of development. So what are these layers and why are they so important? I had a little video of, this is a blastula forming. There's this fluid-filled hole in the middle, and then the cells start to kind of curve around each other and grow. So these 3 tissues end up forming very distinct structures from each other. So the innermost layer is called the endoderm, and these cells will end up giving rise to things like the lungs, your thyroid, which is really important for hormone secretion, as well as your digestive system. And then the middle layer is called the mesoderm, and this is what ends up forming your muscles and bones and your blood and your heart and connective tissues. And then the outermost layer is the ectoderm, and this is what forms both the skin and the nervous system. So we're very interested in knowing how the ectoderm is laying the groundwork for the rest of development. So it's important to understand that all of these cells in these layers are constantly communicating with each other. So cells that develop first guide other cells after them to know what they should be able to do and what types of genes they should turn on or off. So the cell is able to differentiate and know what kind of cell it should become. And without this constant communication, all of the cells would just end up staying stem cells and no structure would ever be built. And in fact, development altogether would just halt since there was no cells telling other cells to continue dividing. So these signal gradients are each unique, and there's tons and tons of known molecules that serve to communicate these different instructions. So I had another question for you guys, if you can remember. What is the name of a single cell, the first stage of life? Is it a zygote, a blastula, a gastrula, the germ layer, or an organizer? Great, I'm seeing some A's. You guys have been, I don't even know, I think this is like 5 for 5. So yes, you are, when you are a single cell, you are called azygote. And then what is the germ layer that's the most important for nervous system development? Maybe this one will trick you guys up. Is it the endoderm, the mesoderm, the ectoderm, the epidermis, or the gut cavity? Nice, I'm seeing C's and that is correct. You guys are doing a great job paying attention. So now that we've kind of formed these 3 layers, we are going to start entering a process called neurulation. So as I said, these 3 embryonic tissue layers inform the rest of tissue development. And from here, we're going to focus on the ectoderm, as you guys correctly just answered that last question, of where the nervous system comes from. So the most critical developmental stage in the nervous system is neurulation. And this is when a structure called the notochord, which is just super important in being able to release those signaling molecules I was talking about. And it signals to the ectodermal tissue above it to kind of fold up and form a U shape. And eventually these 2 sides meld together at the top. And this is the start of your spinal cord. And one side of the tube will end up being where your head forms, and the other side will end up being where the bottom of your spinal cord at the bottom of your back is forming. So how do we get from a neural tube all the way to a fully formed brain? And at this point, we're at about the end of week 4 for human development, just so you guys can think about orienting yourself in that way. But we're very far from reaching the end of brain development. So now that we have the neural tube formed though, and we have some of these neural progenitor cells, so the building blocks to making neurons, we can start to divide and develop them. So once the neural tube forms, the front of the structure starts to develop into the head. And the main developing portion of this is the brain. And at this point, the brain begins to organize itself into the different brain areas, roughly, that it will eventually form. And this organization relies heavily on activation of the body access planning genes, and these are called Hox genes. And control over what genes each cell is expressing allows for the control of development and the eventual control of what kind of neurons and where all of your different areas in your brain are going to end up being. So in general, these primitive vessels, as they're called, lay the foundation for what area of the brain it will be focused on. And the front vesicle is called the telencephalon, and this is what's going to develop into your cerebrum, which is the area of your brain that allows you to think and problem solve and have a personality. So this is a region of the brain that not all animals have or have as developed as we do. This is one reason that humans were able to end up being kind of the dominant species, is this higher order thinking. And under the telencephalon is the diencephalon, which will be a hub for hormone secretion. So these are the things that are regulating different expressions within like testosterone and estrogen. So the things that make people have or animals have male or female sexes, as well as hormones that are important for regulating sleep and like insulin signaling. So you being able to store food supplies, and as well as vision and sensory information is also within this area. And then under that is the mesencephalon, which will be dedicated to motor control, motivation, temperature control. Then there's the metencephalon, and this is primarily involved in forming your cerebellum, which is allowing you to have coordinated movements and have balance. And then finally, the myelencephalon, all the way in the back, controls most of your unconscious bodily functions, such as breathing and heart rate and different reflexes. And this is where the spinal cord connects to the brain. And this is kind of the most primitive of all of your brain regions. So pretty much any organism is going to have a myelencephalon in some shape or form, because everything needs to be able to regulate breathing and heart rate and those baseline actions that you're able to perform. So I totally get that this slide is a little overwhelming, and it should be, because what's listed here are just some of the functions that your brains will end up taking on, and there's a ton of them. So it just goes to show you how utterly complex we have evolved to be, and how that complexity has given us the upper hand in becoming our planet's dominant species. So I have another poll question for you guys, going a little bit back to where that neural tube was forming. And I'm wondering if you can remember how it ends up forming the tube. So are cells only growing so that they end up just forming a hollow tube? Does the mass of cells hollow itself out? Does the top layer of cells grow, that should say up to meet the bottom layer of cells? Do the grow down to meet the bottom layer of cells? Do the sides of the neural tube kind of fold up to meet each other? Or the spinal cord is just immediately developed and you don't need this tube to be the starting factor? Starting to get some answers in. Have a couple D's. And that is the right answer, guys. Great job again. So as we said, you kind of start with a flat area of cells, and then the sides of that plate end up growing up and they end up forming a tube, almost as if you had like a soft taco shell and you were to just kind of curve it up and it met at the top. It's forming in pretty much the same kind of means for that. That's great. And then my next question for you guys is a little bit more open-ended, so I'm interested to see your thoughts. So on the left, we have the brain of a rodent, and on the right, we have a human brain. So I'm wondering if you can see any differences between these, or if you want, like, what are some similarities that you see between them as well? I'll give you guys a second to think about this one. I have the rodent brain is smoother, which means it doesn't have as much information. That is a great observation. It totally is smooth compared to human brains have more folds. And you're right that these folds allow us to increase the surface area of our brain, which allows for more neurons to be there, more connection. Human brain has four defined gyri and sulci. That's great, Leo. Totally. You just put a definition to what Alice said. That's awesome. And then maybe one thing that's a little harder to notice is what's going on with the olfactory bulb. So this is the way that you're able to smell. Do we notice like a difference at all between humans and rodents? we have the rodent olfactory bulb appears much bigger. Absolutely. So, I mean, if you think about the differences between humans, yeah, we can smell, but think about how like rodents and dogs and animals that rely on scent interact with their world, right? Like that's one of their main senses, whereas to us, it's more of an accessory scent. Whereas we have much better vision than, say, a rodent would be able to have. So we dedicate more space in our brain to having visual perception rather than olfactory perception. Awesome. So now that we've laid the groundwork a little bit, we can start to talk about where neurons are coming from and how they're born. So neurogenesis is the process in which neurons are born. And from the ectoderm, you have neural stem cells called neuroblasts, and these are born, and these end up producing almost all the neurons you'll have in your entire brain. So neurons themselves don't divide, and they rely on neuroblasts for their ability to grow and make more of themselves. So it's important to note that you're born with basically all the neurons you'll need for your whole life. So these cells are extremely long-lived, especially compared to, say, a skin cell, which replace themselves about every 2 weeks. And this makes protecting these neurons really essential, and why we need to take brain injuries so seriously, because there's not much regenerative properties of your brain as you go on through your life. And to start out, neuroblasts divide again and again to make themselves able to meet the demands of producing so many neurons. And these are called symmetrical divisions, since the cells are dividing equally, and it results in cells that are going to be the same cell type. So once there are enough neuroblasts, the production of new cells switches primarily to producing neurons, and then some glial cells, which will support those neurons. And then later on, the focus shifts to just producing glial cells to support all of the neurons you already made. So then once neuroblasts begin dividing to produce neurons in glia, we call this an asymmetrical division, since there's two different cell types being produced. And neuroblasts continue dividing, and the other cell, like the cell that was made that's not going to be a neuroblast from the asymmetrical division, has to migrate away from this area, because the neuroblasts are just going to continue to divide in the same place. So those cells have to be able to move away from that area and travel to wherever their final destination is going to be. So the new glia or neuron hasn't completely differentiated yet, meaning it hasn't taken on its permanent cell identity. And it won't do this until it reaches its forever home. So during neurogenesis, neuroblasts end up producing way more than we need in our adult life. I'm pretty sure in like insects, they produce 3 times the amount of neurons that they end up needing. And in mammals, we end up producing 2 times the amount of neurons we're actually going to need. So there's a ton of selective cell death that occurs after all of these cells are made, and because it's better for the brain to overproduce the amount of neurons it's going to need than produce too few and end up having huge problems. So how do neurons in glia migrate? Like you can see this cell is on the screen right now. So basically there's these cells called radioglia, which act as a stable structure for the migrating cells to wrap themselves around, and they shimmy up, kind of like if any of you or your parents are into gardening and you have plants, you can give them a little pole and they're able to kind of climb up, and it gives them something to hang on to. So once these cells reach their final destination, they just hop off. And at that point, they're able to receive those chemical cues from surrounding cells that tell them what kind of neuron or glia cell they should end up becoming. So each wave of migrating cells actually migrates past its predecessors, and they have to settle further out within the brain. And this is referred to as an inside-out development, where the layers that are closest to where division is occurring from those neuroblasts are the oldest cells, and then the closer you get to the surface of the brain is where your youngest cells are. And there's one exception to this rule, that one of the first layers to be developed is actually the very top layer of your brain. And this is because those cells tell all of the new cells that are being born to migrate up. So it gives them a sense of location when they're born and where they're supposed to go. So I have another poll question for you guys, so feel free to enter it in the chat. So I'm wondering if you can remember what a neuroblast is. So is it a differentiated neuron, meaning it's taken on its final cell fate? Is it a cell that makes neurons and glia? Is it a guidance structure for migrating cells? Or is it the outermost layer or the innermost layer of developing cells? We have a couple answers. We have a couple B's. Does anyone else have any thoughts? More B's? Well, for those of you who answered B, or maybe you were thinking of it and you were just a little unsure to enter it in the chat box, you're totally right. So neuroblasts are those cells that are the main producers of neurons and glia. So now we're going to get into one of what I think is the coolest parts of development, which is outgrowth. So at this point, we have gotten to the time where we have made a bunch of neurons and they have decided what kind of neuron they need to be. And they're doing that through the communication of different like guidance molecules and chemical cues. But so far they haven't made any connections yet, right? Like that's like the most important part of neurons is they're able to form connections with other neurons so they can communicate. So neurons have these long projections called axons, and they reach out and they try to find targets. And a growth cone is what's at the very tip of the axon. And this is kind of like a baby axon. Like these are what's going to form those axon terminals that are going to release the chemical cues, those neurotransmitters. So they have these finger-like projections, which search around for chemical guidance cues, finding out where they should connect to. And this is a really amazing feat because some of the axons in your body are actually ridiculously long. We have some that stretch all the way from the bottom of your spine to your big toe, which can be for some people like a meter. Or imagine like the tallest person in the world, right? Like that's a super long distance for one axon to have to travel. So once they find their target cell, the growth cone ends up turning into axon terminals, like you saw in the beginning, and that's where they form these synapses with the dendrites of another neuron to be able to produce neurotransmitters waiting to be released in response to an electrical cue. So that brings us kind of full circle all the way back to the beginning of the talk for how neurons communicate. And once neurons are able to start communicating, that's when there are major modifications in cell communication. They start to form pathways with each other. And after birth, there are major periods of learning and brain development. And the brain doesn't finish developing until you're around 25 years old. And even then, like you're still able to learn, so you're still able to make modifications and add new connections and lose connections throughout your life. And I also want to mention that this was just kind of dipping your toe into neurodevelopment because there's so much more complexity to it, and we could talk forever about how important genetics are in neurodevelopment and the different behaviors that form. We didn't even touch upon postnatal development at all, like all of the learning that happens when you start out as a baby and you have to learn how to walk and talk and exist in this really complicated world, right? So this is just kind of the beginning, and I hope I piqued some of your interest. And before we finish up, I just wanted to give you guys a little taste of what I am really interested in and what I'm currently studying. So in my research, I studied the development of glial cells, which are those neuronal support cells. And I mentioned before as I mentioned before, sorry. And to do this, we study flies. So we use the same kind of flies you probably see in your kitchen, just hovering over like old produce. And yes, they totally have brains. So we study how their brains develop. And to do this, we use larvae, and they look like what you guys see on the left-hand side there. And fly larvae only have about 100,000 neurons in their brain, which probably sounds like a lot, but an adult human has over 100 billion. So they serve as a much simpler model to be able to study the different neuron interactions that are happening. So flies start off as an egg, and then that fertilized egg develops into an embryo. Then they enter larval stages. The one I'm most interested in is L2 to L3. This is where there's a huge boom of both neuron and glial growth and development and birth. And then next they enter pupal stages. So you probably know this in the context of like caterpillars turning into a butterfly. They have to form that cocoon. So there's a huge body reorganization that happens as they go from a larva into an adult. And that whole process from embryo to adult only takes like 10 days, which is super fast, and it makes lab experimentation a breeze. I have tons of stocks of just like growing larvae, and they're super happy to just kind of sit in a little pile of like goop that's like sugar and different like amino acids and stuff like that. And even more powerful than their speedy development is the ease in which we're able to genetically manipulate the different cell types within them. So since we're interested in the brain, we're manipulating neural cell types, and we can completely remove different proteins that these cells make, or we can insert a completely new protein into them, including fluorescence. So we're able to take pictures of them under a microscope. And they also have their like outside skin, which is their cuticle, is clear when they're larval stages. So we can image their brains while they're still alive. So you can take time-lapse photos and things like that. And they have a lot of conservation of a lot of the same signaling molecules and signaling that's happening in these cells are completely conserved within mammals. So it's really easy to apply what we learn in flies to humans. So I study cortex glia, which is the little blue dot you see in this picture. And then the green are all of its processes, and they're wrapping these red circles, which are neurons. And cortex glia are really important in supporting neurons within fly brains. So they're still a glial cell type. They're working like microglia and astrocytes do within the mammalian system, that they are trophically supporting these cells. So they're feeding them, giving them nutrients. They're also structurally supporting them with these processes wrapping around them. And we know that they are able to kind of clear dead and dying cells away to keep the brain happy and healthy. And each cortex glia wraps about 20 to 80 neurons, which is quite the range. So there's a much fewer cortex glia than there are neurons. And they send out like this extensive kind of webbing that you see here to wrap around them all. So I'm trying to figure out if cortex glia have multiple nuclei. So they have multiple nucleuses within one cell, and if that's helping them to be able to care for the neurons that they're working to surround. And to do this, we're able to have a cortex glia that starts out green, and then with exposure to a certain laser, it's able to turn red. And this color diffuses throughout the cell, so it ends up showing us where its boundaries are, and where it can count how many nuclei are within one cell. So understanding how these cells are functioning can give us insight into how our own mammalian glial cells are working to take care of all of the neurons around them. And with that, thank you guys so much for listening and for answering my questions that I was asking you. You paid really great attention. So now I'm just wondering if you guys have any questions. I would be more than happy to answer some of them.
Yes, so before we get to questions, and feel free to put your questions just right into the chat box, but I'm going to launch a poll. If you all can just answer the 2 questions here first, and then put your questions into the chat box. And I know Hannah is very happy to answer any questions you might have for her. Just give it a moment, give everyone a chance to answer this poll. Give it about another 10 seconds. If those of you who haven't answered yet, hopefully you see the poll. Okay, I'm gonna close up the poll. Thank you all for doing that. So if there are any questions, Leo says he loves the cartoons.
I know, it's one of my favorites. I've started ending all my presentations with it. You gotta love a corny joke, right?
We'll give it a second for you to, if there is a question, don't feel like you have to ask a question, but you have a wonderful opportunity with a PhD student who studies this interesting topic. But if there are no questions, then let's just thank Hannah for taking the time to share her work with all of you.
Oh, we have one question.
Oh, we do have a question. Yeah. So you said that neuroblasts produce neurons and glia. They are a single cell that can form 2 different cells that go on to do much different things. How does that work?
Yeah, so there are, like I said, these like signaling molecules within neuroblasts that while the cell is dividing, these some of these like chemicals end up picking a side of the cell, so they have like a polar distribution, and then depending on where they end up when the cells completely divide influences the identity that it's going to keep. So that's how a neuroblast can make multiple different types of cells. Like you could put chemical A, B, and C in one, and that'll influence what it wants, or it could put molecules D, Q, and A in another one, and that could make a completely different cell type. And then it's able to keep its own identity after dividing based off the distribution of molecules that happen during division. Yeah, it's super crazy.
Cool.
You're even like when you're born, when you start off as a single cell, you actually get a certain distribution of those signaling molecules within that cell from your mom. And this is called maternal contributions. So you're already set up to start making decisions on cell fate from that first instance of being a zygote.
So last call for a question or 2. Leo, somewhat different. What causes headaches?
Headaches can be caused by a variety of things for sure. Most of the time it's due to a change in the volume that your brain has. So either having your blood vessels in your brain be dilated, so they become larger or constricted. And that's kind of what causes people to have headaches when they're withdrawing from things, especially caffeine. As I'm sure the adults in the room know, you get to a certain part of the day and you start to have a headache. So yeah, different types of concentrations in your brain within those blood vessels in regards to volume can cause you to have a headache. It's kind of like the response to an increased or decreased pressure within your brain.
I like these questions that are being asked. So Alice asks, how do you justify experimentation on living beings? Where would you draw the line?
That is an excellent question, and one that is super important within especially neuroscience, since there's a lot of animal lethality that has to have to be able to experiment and answer these questions about the brain. It's really important that when people are entering neuroscience, they understand what the consequences are for what they're doing, and they respect the fact that something had to give their life to be able to progress science. That being said, there's a ton of different model organisms. Some of them are easier to justify, at least I would say. Like right now I work with flies. One reason I switched from birds to flies is because it did, it weighed on me heavily that I was having to like take the life of birds, but it was a lot easier for me to do larvae. But yeah, some people work on primates, which is really hard. And I know people that have had to sacrifice animals that they've worked on. And it weighs heavily on them because they form relationships with them since they're like so complex. But one thing science does to try to decrease the amount of life is depending on the organism, like in flies, the amount of animals I'll have in one study is much higher than the animals that people would use if they were using monkeys or even rodents. So you try to use, there's different methods in science to try to up the amount of data you can gain from one organism, but it's definitely a drawback. And now people are working with stem cells. So you're able to just take a culture of some type of organism and grow cells in a plate, which has made it a lot easier and has reduced the amount of life that we have to sacrifice for the progression of science. So that's a great question. And there's entire institutions within universities and within the government that kind of set up the standards of life and what you can and can't do within experiments and to reduce pain and things like that in the process. So if you're interested in that, in animal advocacy and science, that's a whole realm of a science career that you could have.
So Amelia asks, how is it decided which neurons die and which end up surviving during neurogenesis because too many are formed for all of them to make it?
That's a great question. It depends on the organism. Some organisms, it's really regulated, like within like worms, you can characterize exactly what neurons are going to be born and then exactly which ones are going to be able to die. Like they'll have names like 1A, like 200C, things like that. Within more complex animals like humans and mammals, it's not as characterized. A lot of it has to do with what cells were older and had more time to form stronger connections, and which ones are just kind of hanging out and they totally couldn't be gotten rid of. And a lot of that decision making happens within neurons. Even like right now in our own brains, there's certain like memories and connections that you form that we just don't really need anymore. So your brain tries to be efficient and it kind of prunes and deletes those connections, but you still have the opportunity to gain them back. But yeah, that's a great question. It's not as planned as you might think, but definitely just cells that are able to establish themselves more have a better chance of being selected to stay there forever.
Well, again, great questions that have been asked. So if there are no more, let's thank Hannah again for her time, for sharing her work. This was fantastic. And hopefully we'll see you all back next week. We have, it's on, I think, humor in babies. Should be really interesting. So come back, check it out. And everyone have a good rest of your day. Thank you for joining us.